Hi everyone. Today I want to talk about graphing in science, specifically how we graph in the biology class that you're going to be taking. So first, in order to talk about making good graphs, we have to discuss really basics about a good experiment. So in a good experiment, there's always a control group present. You're only changing one variable in each experimental group, although in your total experiment you might have multiple experimental groups. You have multiple trials, you try to minimize bias, and usually have peers review your work. Today we're going to focus on the things that actually affect your graph more. So we're going to discuss our different groups, control groups and experimental groups, and we'll discuss then how they look on a graph. Generally, guys, in any experiment, we are trying to compare two variables and trying to see if there's a relationship between these two variables. We call one of them the independent variable. The independent variable is the variable that the scientist changes. This can be a qualitative or quantitative variable, and they're looking to see if there's some effect on a dependent variable. The dependent variable is the factor that the scientist is actually measuring. So we look at this initial experiment here. The independent variable is actually just time. In a lot of cases, the independent variable is something else. And the dependent variable is the condition index of the iguanas. So again, the independent variable is the factor that the scientist changes. The dependent variable is the factor that they're measuring. The dependent variable is always on the y-axis of a graph. The independent variable is usually found on the x-axis, but not always. I'll show some examples where the independent variable is not on the x-axis. Okay, whenever you're looking at a good experiment, there are always a comparison of two major groups, a control group and an experimental group. The control group doesn't have any treatment done on it, and the experimental group, you are changing the independent variable in some way. So I'd like you to take a moment, pause the video, and look at this graph and decide what the control group is. Make sure you read the figure title and examine the labels on the graph. Okay, whenever you're deciding on the control group, you should first decide on what the variables are. The dependent variable, in this case, is the stomach contents mass. And there are two really independent variables. We're looking at the snout vent length compared to the stomach contents. Then also we're looking at different time periods. So which one is actually in the experiment that we're comparing to? Well, in this case, they're really comparing between the normal event or normal time after 1991 and 1992 to this environmental change that occurred during 1991 and 1992. So the control is actually after 1991, 1992, and so. In some experiments, there is a positive and a negative control group. The negative control group is an experimental group that should not change in the dependent variable. A negative control group is used to make sure that there's no unintended factor in the experiment that could give you a positive result. So if you're doing experiments and you want to see if something's occurring, and you get everything's occurring, well, you don't know if it's because of the factor of the experiment unless you have a negative control group. A positive control group is where a treatment should cause a change in the dependent variable. And this is used to show that the testing procedure does work. So for instance, if you had experimental results that showed everything was negative, well, is that because the experiment was flawed, or is that because they actually didn't show a positive result? Over to right here, this is an example of a negative and positive control used in an experiment to test for protein. The protein test is called a Bayer-Retz test, and it is used to show that there is proteins present. A purple color shows that there's proteins present. A blue color shows that there is no protein present. What do you think the negative control would be in this protein test? The negative control should be water. Water will not contain any proteins. And so you can prove that the blue color shows that there is no proteins by using a negative control, which is water. What do you think the positive control should be? Positive control should be a known protein source. For instance, egg protein. If you use a known protein source, you should see this change to a purple color. If there was no change to a purple color, what would that tell you? If there was no change, then you know your experiment is somehow flawed. Most likely in this case, the Bayer-Retz solution is somehow contaminated, or you're not using bio-red solution for some reason. In a good experiment, there are other variables that are held consistent and are not changed throughout the experiment. And there's two examples of those, and sometimes students confuse these. The first are controlled variables. Controlled variables, a lot of times students confuse with the control group. Remember, the control group is something you are comparing to an experimental group and looking at how the dependent factor changes. A controlled variable is something that you are not changing in an experiment. For instance, if you were going to grow plants in an experiment, you would use all the same type of soil. If you use different types of soil, you'd be changing the experiment, and it could be the soil type that was actually affecting an experiment, rather than, say, the amount of fertilizer you might be testing. Constants are very similar to controlled variables. Constants are factors that are not controlled by the scientist, but they remain the same throughout the experiment. For instance, if you're growing plants outside, a constant would be the amount of sunlight they receive, as long as you are growing them in the same location. Okay, so there are different types of graphs. Line graphs I want to mention and highlight. They're the most commonly used in biology. If you're trying to figure out what graph to use, start with thinking, can I make this a line graph? And then move on to the others if the line graph doesn't work. Scatter plots, we'll discuss that in a moment. Bar graphs, histograms, and circle graphs are other types of graphs. Okay, so we're going to start with line graphs. So in a line graph, you plot the information with points and you show a relationship with a line. In a line graph, there is always a quantitative variable on the x-axis. In this case, the quantitative variable is time. In a line graph, time doesn't have to be the only quantitative variable on the x-axis. In a previous graph, we saw that snout length was the quantitative variable on the x-axis. Usually when we make a line graph, we draw out the points and we try to show a relationship using a line of best fit. To draw a line of best fit, you plot out the points, and then usually if we're drawing this out with a graph, we just take a ruler and you try to fit the line in between all of the points. There are processes on data sheets like Excel or Google Sheets that you can use to also find a best fit regression line. There also may be other mathematical models that can be fit towards points, but most often we'll use a linear relationship. Really quick too, did you guys notice something I'm missing from this graph? That's right, I'm missing a title. What is the independent variable in this experiment? So the dependent variable is the percent change in mass. You'll notice for each of these, what this experiment is showing is you take chunks of apple basically and you put them in different concentrations of sucrose. And then you look at their change in mass. In experiments where there is very little sucrose, water comes into the apple and they gain mass. In experiments with lots of sucrose, they lose mass. This is showing them being put in different concentrations. They're actually put in 0, 0.2, 0.4, 0.6, and 0.8 molarity sucrose. But the independent variable really that we're looking at here, that we're experimenting to see the difference in, is the difference between Macintosh apples and Honeycrisp apples. Macintosh apples actually cross this line at a higher point. This is where they're at equilibrium, where there's the same concentration of solute inside or sugars inside the apple as there is outside. So in the Honeycrisp apple, they have a concentration of about 0.45 molar sucrose, and in the Macintosh apple, they have a 0.30 molar sucrose. The next graph is a scatter plot. Scatter plot starts off the same as a line graph. However, in a scatter plot, you don't usually see any mathematical relationship between the independent and dependent variable that's clear. So you don't draw it. You can see a sense of the difference in data, but you don't see a linear relationship. For instance, you can see all of these squares up top here generally. That's showing lower intertidal samples. And so in general, in lower intertidal samples, at any sea temperature, the height of algae is taller. Bar graphs usually show qualitative data on the x-axis. So you don't have a quantitative data on the x-axis. Use a bar graph because you can't show a relationship between two things. These are showing genotypes, and we have a homozygous AA, homozygous GG, and this heterozygous. There's no way to show an in-between of this. There's no like half G in between. So we have to use a bar graph to show the difference between these three pieces of information, these three genotypes. Take a look at this graph real quick. And what do you think the control group is? It's actually not listed here. But what do you think it represents? Okay, the next type is a histogram. Histograms are often confused with bar graphs. Histograms, unlike bar graphs, often show quantitative data on the x-axis. However, they show intervals of data. Usually histograms in biology are used to show populations and how populations change. In this graph showing ages, there isn't a single age. It isn't like 21 that we're looking at a frequency of. Instead, we're looking at a range of a couple of years. So the second graph is showing the amount of calcium phosphorus product in intervals in patients with hyperparathyroidism. Circle graphs or pie charts are least used usually in biology. They're often used for a lot of survey data. But this is a great example of a biological circle graph. This shows the percentage of different DNA sequences in the human genome. Really quick, what's really interesting about this is it shows that very little, 1.5%, is actually used for coding proteins. Most of the rest of this information here, a lot of it is repetitive and used for regulating these genes. All right, so what are the components of a good graph? First off, you need to make a good graph on graph paper. Now, in some cases, we're going to use electronic graphs, like you can make them on Excel or Google Sheets. But in a lot of cases, you need to start with graph paper. Now, some students will ask me, where do I get graph paper from? A lot of times, if we're in class, I provide graph paper. Usually on documents, if it requires a graph, I'll try to include graph paper. But if I forget or if there is no graph paper on the document, you can easily find graph paper online. Search for graph paper. Also, a lot of drawing applications, like Microsoft Whiteboard, have a graph paper background that you could use as well. So you could draw electronically on the graph paper. You should have your axes, your x and y axes. The axes should be labeled, and units for each of those axes should also be labeled. You should properly scale the graph so that it fits most of the graph paper, or approximately 2/3 of the graph paper at least. And there should be a title with a figure label at the bottom. The title should be descriptive, and it should never be, in biological graphs, a versus title. Like it should never be nitrogen versus growth in plants. They're not competing with each other. If you were going to draw a graph of how much nitrogen fertilizer affected plants, you would say really the growth of plants and label the species really of plants in different amounts of nitrogen. Okay, so let's take a look at some data and let's discuss how to draw a good graph. Okay, so as an example, we're going to graph this data here. So before we start the graph, you should determine what your different variables are. So what is the dependent variable in this experiment? The dependent variable is the length of stem of these fast plants. What is the independent variable? The independent variable is the amount of ammonium phosphate. What's the control group? The control group is where the independent variable has 0 grams of ammonium phosphate. In fact, this is a negative control group. We don't expect it to change the experiment. So now that we have identified our variables and our control, let's start our graph. So first, we do need a piece of graph paper. Then we need to determine what's on the x and y axis. So what goes on the x-axis? So in this case, it is not the independent variable. It's not the amount of ammonium phosphate. It is actually time in this case. The independent variable we're going to show with actually multiple lines. Now the dependent variable always shows up on the y-axis, so that is the length of the stem. You'll notice on my x and y-axis labels too, I have included the units. Do not forget those. And then finally, I need to show my independent variable, and to do that, I'm going to use a key, and I'm going to use 4 different lines. I will color code them too as well with these different colors. Okay, the next thing I need to figure out is the scaling of the graph. So figure out the scale of the x and y axis. I want my graph to fit in this graph paper, at least 2/3 of the graph paper it should fit. Okay, so let's look at our graph paper. For the x-axis, we have 10, 20, 30 lines. And we need to be able to figure out how these 30 days fit into those 30 lines. So whenever you're figuring out scaling, what you should do is look at the range of your data and look at the number of lines. Now, we have actually a range of 0 to 30. I didn't include 0 days in the data because this is using fertilizer and germination. It starts with a seed. The seed is not going to have any stem length. So we're starting actually from 0 days with a length of stem of 0. Okay, so to figure out scaling, you take the range of data, in this case 30 days total, and you divide by the number of vertical lines. So there's 30 vertical lines, that gives 1 day per line. Then I write down my labels. Now don't label every single line, I'm going to label every 10 lines. By the way, one quick thing too, a lot of students will trip up and they'll write down just these labels and try to put those on markings. That would be a mistake because the difference between these intervals changes. I have a 0 to 5 day actually for this first one. That's a 5-day interval. 5 to 10, that's a 5-day interval. But then I have 10 to 20, that's a 10-day interval. If I did that, that would warp my graph and that wouldn't show the relationship correctly. So instead, I want to keep that consistent. Have 1 day per line. 5-day data is going to go right here, and then 10 here, and then 20 and 30. Okay, now on the y-axis, you need to look at the range again. So what is the range on the y-axis? So the range on the y-axis, if you find the maximum here, it's 19. And again, we're starting from a length of 0, so 19 minus 0. There are 40 horizontal lines, so we take that 19 divided by 40, and we get 0.475 centimeters per line. 0.475 centimeters per line would be horrible to work with. So we want to actually round up to an easy to use number. It does mean our data isn't going to go to the very top, but it does mean it'll be easier to work with and write, and people will understand it better too. So we're going to round that to up to 0.5 centimeters per line. By the way, we don't round down because if we round down, what will happen is our graph will have to go above the graph paper. And I see lots of students often will draw in extra lines up here, and then I will take off points for them drawing in. Use the graph paper that's up here, okay, and only go to this maximum. Don't add graph paper to it. Okay, so 0.5 centimeters per line. Again, I don't want you to mark each line here. I want you to pick major marks. So I'm going to pick every 10 lines. So 10 lines times 0.5 centimeters means 5. And then the next one would be 5 from that, so 10. 5 from that, 15, and then 20. Okay. If you wanted to put in between marks, you could put one right here, like 2.5, 7.5, if that made you be able to keep track of it a little bit better. Well, actually, we forgot one other thing, and make sure you don't forget this thing. We need to draw a title for it. So the title should always go on the bottom. It should have a figure label. So this is figure 8, and the title should be a description of what the graph shows. Don't be afraid to make a whole sentence in your figure title. You will have noticed, if you look back at the old slides that I showed that were from published papers, they had long statements describing what was happening in the graph. There was never a verses in there, so don't use verses. Describe what it is. Also, if you're using an organism, make sure you include the genus species of that organism. Okay, so the next thing we need to do is plot our points. So if we start with 0 grams of fertilizer, at 0 days, there was 0 stem length. I'll plot that out at the 0 points. Then at 5 days, there was 2 centimeters of growth. So we look at, count at 1, 2, 3, 4, 5, and we go up 2. Now each line is ½ a centimeter, so 1, 2. There's my points. Okay, and then I plot out my 10 days, which is at 4. I plot out my 20 days at 6, and then my 30 days at 10. Once I have my points plotted, I can draw in my best fit line. To draw in your best fit line on a piece of graph paper, use a ruler and try to fit in the line between all the points. So here's my best fit line. Okay, and then I'm going to plot out my other points for my 2.5 grams. I'm going to do that really quickly. I draw a best fit line. Do the same thing for a 5 gram. We'll do that really quickly again. Draw a best fit line. And then I'm going to draw the same thing for the 20 grams of fertilizer. And draw a best fit line. First, before we move on, you should be able to make a conclusion from this graph. What can you see in this graph? Pause it and take a look at what the graph data shows. You should make the conclusion that 5 grams of ammonium phosphate shows the highest amount of growth in the fast plants. In fact, you could compare these numbers here and look at, or the slope, and look at the difference in rate of growth. Okay, one real quick thing again to remind you is lines of best fit. You should not extend the lines past the points. And that's because you don't actually know what's going to happen after the points. Now, obviously, 0 is the earliest point. You can't extend beyond there. But after 30 days, you can't figure out what the growth is actually going to be. In fact, fast plants after 30 days, they're pretty much mature and they don't continue to grow much at all. So making a conclusion that they continue to grow forever by drawing your line past 30 days in that same upward slope would be incorrect. Also, did you notice the point that I misplotted? Take a moment, pause the video, and find the point that I misplotted. So it was this one up here. I plotted it 1 below 20. Sometimes when you're working fast, guys, you might forget your scaling. So I thought really quickly, underneath 20 is 19. So I plotted my point at 19. But remember, there is 0.5 centimeters per line. So I should have put it, instead of there, should have put it right here. Okay, I just want to really quickly review scaling of axes. Axis scaling is one of the things I see a lot of younger students make errors with and have difficulty with. To find the scale of an axis, remember to find the range of data, and the data doesn't have to start from 0 like it did in my last graph. Divide the range of data by the number of lines across the axes, and round the number up to an easy-to-use whole number or decimal. So for example, here's some data. The other night I was watching a video on different large cats and how they were different from each other. And one of the things mentioned was the size. So I looked up the female's average mass of each of these large cats. And now I'd like to make a plot of them. So what type of graph should I use in this case? Well, in this case, because the independent variable is the large cats, the best graph for this would be a bar graph, because there's no quantitative variable here as the independent variable. So let's draw a graph real quick and figure out our scaling. So on the y-axis, of course, is always going to be the dependent variable, female average mass. And in this case, the x-axis will have the independent variable. So let's put the large cats, or the independent variable, on the x-axis, and put the dependent variable, female average mass, on the y-axis. Now you should see really quickly that there are 3 basic blocks here that you can put your large cats in. So scaling for the bars is going to be pretty easy. For bars, in a bar graph, you usually want to leave space between them, and you want to make sure that the bars are equivalent size. We'll show that to you in a second. The average mass is where we really have to focus on in scaling here. So what would be the scale for the y-axis? Again, we have 40 lines. We have to see how to split that with the average mass. Now, in general, you want to look at this range and figure out the range and divide it by the amounts of lines. So if you take a look, it's 142 minus 40, and then we divide it by the 40 vertical lines, and that gives us about 2.55 kilograms per line. Okay, so I said in general we want to round this up to an easy-to-use number. So we're going to round this up to a whole number, 3 kilograms. Now that's also going to allow us to do something else. I do not want to start this graph at 40. 40 would mean that Cheetah's bar would show up right on the bottom and would look to most people really quickly, not paying attention to the scaling, that the cheetahs weighed nothing. So instead, we're going to make the range so that cheetahs show up as a bar. So instead, I'd like to start with a 30. And then if I use my 3 kilograms per line, I have plenty of space to still fit in the tigers. So 3 kilograms per line, there's 10 lines, right? So that'd be 30 for each major mark. So 30 plus 30 is 60. 30 plus 60 is then 90 for the next one, and then 30 plus 90 is 120, and the last one will be 150, giving us plenty of room for the tigers. Okay, then I can plot out my independent variable and each of the bars. Again, you'll notice that the bars are equivalent widths, and there's some spaces between the bars. Oh, don't forget your title. A lot of students always forget it, and in either biology class, you'll lose points if you forget your title. The title is very important. Okay, in some graphs, you're going to see standard error bars. Especially in AP Biology, you'll see standard error bars on almost all graphs shown in the AP exam. Standard error is based off of standard deviation. Standard deviation is used to show the variability in data. Standard error is calculated by actually dividing it by the square root of the sample size. And it actually shows how far the data is likely away from the mean. What that means really is that it tells you how significant the data is around the mean. Okay, so again, standard error you calculate by taking standard deviation and dividing it by the square root of the sample size. Standard deviation, we actually have to calculate by looking at each of the individual samples, subtract it from the mean of the samples, and then square it. And it's the sum of all of these. So you have to go to the next sample, subtract it from the average, and square it. The next sample, subtract it from the average and square it. And then all of those things divide by n minus 1 and take the square root. So again, standard deviation shows variability in the data. Standard error shows how far the data is likely away from the mean. Most of the time, we're going to see graphs that look like this in AP Biology that have a line graph with standard error bars. And the most important thing you should remember about standard error bars is if they overlap with another point. That means that the data within that standard error, if they overlap, like if we look at this point, and they overlap a little bit, or even close to each other, really. These ones, this is maybe a better example. These overlap a lot. That means that there's no difference, really, between these 3 points, really, if these standard error bars overlap. There's no significant difference in the data. However, if they do not overlap, if you look at the difference between these standard error bars and these, it's a huge difference. This mean is definitely significantly different from this. Okay, so most of the time you will see standard error bars. Make sure you interpret the bar and look to see if they are overlapping. If they're overlapping, there's no significant difference. If they're not overlapping, there is a difference, and this may actually mean something that's important. While making this presentation, I actually mistakenly put this graph up here first until I looked at it closely. Sometimes scientists will choose either standard deviation or standard error to publish. This is actually using standard deviation. Standard deviation, when you interpret a graph, is different from standard error. How do you think the bars would be different if they were standard error bars? Standard error bars would be a lot smaller. Now, since these are standard deviations, it shows you that there's a lot of variation in this data, but there is still actually more significance to some of these changes if we looked at standard error. Of course, either way, if you interpret these bars to each other, standard deviation or standard error bars are going to show you that these really are different, especially this point up here is significantly different from any of these time points. So something happened here. The air temperatures increased here, and you might wonder why that is. Okay, so that's all I have for graphing today. Please use this in the future if you have any questions. Make sure you review your independent and dependent variables. And anytime you look at a graph, think about those things. Think about what the independent variable is, what the control is in the experiment. That's all I have for now, guys. I hope you're staying happy and healthy. Bye.